Hello everyone, welcome back to my channel. Today we'll discuss the histology of the liver. Liver is covered by a capsule connective tissue structure that is called Glison's capsule. This fibrous capsule sends septas and divide the liver into hepatic lobules. Liver histology can be studied either in the form of a hepatic lobule or portal lobule or portal acinus. So we'll discuss that first, hepatic lobule. Each hepatic lobule is a hexagon in shape. We first make an outline of a hexagon, but don't make it too dark because this fibrous tissue is very thin and it is very thick in case of pigs. In humans, the fibrous tissue is very thin. The center of the hepatic lobule contains the central vein, also called terminal hepatic vein. So first we have drawn the terminal hepatic vein, then we have to draw the hepatocytes radiating from the central vein to the periphery. These hepatocytes are single cell thick. They'll be branching and anastomosing with each cord. So in between the hepatocyte layer, we have a space that is filled with the blood, that the sinusoids will be there. So hepatocytes will be polygonal in shape. It almost resemble like a cube with 6 surfaces. 2 of its surfaces will face the sinusoids, whereas the other 4 surfaces will face the neighboring hepatocytes. It's like a 3-dimensional. You can imagine a wall with a brick. So it's a single brick layer. So if you see from the top, it is a single brick layer. If you see from the side, you can see the wall next to another. So the other sides of the brick. So if you imagine a 3D dimension, we can see the hepatocytes. 4 walls facing the hepatocytes, 2 to the neighboring, 2 to the bone below hepatocytes, and 2 faces, the 2 sides face the sinusoids. Once they reach the fibrous tissue surrounding the hepatic lobule, that is, we have a connective tissue which is perforated, that's called limiting plate. The hepatocytes can pass through this limiting plate and sometimes can be continuous, appears as continuous with the hepatocytes of the neighboring lobule. So I'm just shading the background, so easy to draw the sinusoids. The sinusoids are nothing but the dilated vessels. The vessels in the portal canal, they'll give a branches radiating in between the portal canal, radicals. Those vessels will pour into the sinusoids. So sinusoids will get blood from portal vein also, which is having a pressure of around 8 millimeter of mercury, and hepatic artery, which is having 90 millimeter of mercury. In the sinusoid, both hepatic artery and portal vein blood will mix up and move towards the central vein. Each hepatocytes have a centrally placed round nucleus. Some of the hepatocytes can have 2 nucleus, binucleated. So keeping the famous nuclei for the central vein and centrally placed nucleus, in some of the hepatocytes you can keep 2 nucleus. Functionally, the hepatocytes are very important. They secrete bile and also many proteins, most of the proteins, and many are also responsible for the hormone productions, the activation of the hormones like vitamin D, A and K. It's also storage sites for the vitamin A. The ito cells will be present in the space of Disse, which is present between the endothelium of the sinusoids and the hepatocyte. There is a small space between the hepatocytes and the sinusoidal endothelium. That space is called the space of Disse. Sinusoids will be lined by endothelial cells and also cell derived from the mononuclear phagocytic system, that is the Kupffer cells, which acts as a phagocyte, and lot of cellular processes will be there. These cellular processes intermingle with the cellular processes of the hepatocytes also, and they are the one which fill up the space of Disse. In the portal canal, it is surrounded by a connective tissue. Between the connective tissue of the portal canal and the hepatocyte surrounding, we have a small space, that is the space of Moll. And from this space, the lymphatics of the liver will start. So the bile canaliculi they'll be present between the hepatocytes. They are the separation of the hepatocyte epithelium, and when they come towards the portal side, they'll drain into canal of Herring. The canal of Herring will drain into the bile duct, which is present in the portal triad. So the bile flow is centrifugal in direction, whereas the blood flow in the sinusoid is centripetal in direction, flowing towards the central hepatic vein. So hepatic lobule is nothing but a venous unit. So it is the parenchyma of the liver drained by one hepatic vein, which is roughly hexagonal in shape. So I'm just keeping the flattened epithelium for the sinusoidal endothelial cells. Somewhere here and there you can give some cuboidal epithelium representing the Kupffer cells, but it is hardly difficult to identify in HND 10x view. So now in the portal triad, it can be a misnomer because apart from the bile duct, hepatic artery and portal vein there, we also have lymphatics which begins in the liver there. So basically we have to show some three structures there in the portal triad. Hepatic artery, so similar to the how you draw the artery, round structure. Portal vein is histologically similar to the vein, so collapsed lumen. Whereas a bile duct will be lined by cuboidal epithelium. So you have to show the small duct lined by cuboidal epithelium that represent the bile duct. So that's a hepatic artery, portal vein with a collapsed lumen, and another is a bile duct showing the cuboidal epithelium. Those are the portal triad structure, and there is a small connective tissue surrounding the vessels. So basically you should know about the portal triad structures: space of Dissay, space of Moll, sinusoids, how the draining area, the endothelial cells for the artery and the vein. The radicals of these, the branches from these artery, hepatic artery and portal vein, they run in this limiting plates in between this thing, and then they pierce the limiting plate and pour the secretions, the blood, into the sinusoids. This is the histology. Thank you for watching.